Okay, hello everyone. Welcome to our short channel analysis in the Brain Analyzer toolbox. We'll have Dr. Hendrik Santosa presenting today. He is part of the Hubert Lab, as you already know, and they have recently published one article about the short channels, which he will present to us today, and also talk a little bit about how to use the short channels in the Analyzer toolbox. I was privileged to have a chance to visit this lab a while ago during my PhD studies, and I can just attest the high quality of the work that has been done there. Just before I pass on to Dr. Hendrik, I will just talk a little bit about general information for this webinar. So I'm Maria Delia. I'm a scientific consultant here at NIRx, and I also have in the backstage Dr. Milan Harry and Amy Young helping us to run the show. You are muted. Any question is welcome at any time. Please just use the GoToWebinar panel. And all the content will be available in our website, including the slides. So feel free to get it just after the webinar. And if you have any further questions, just write us an email at consulting@nirx.net. As you already know, we are NIRx. We are an fNIRS solution provider. We have offices here in Berlin, where I'm sitting, and also in the US. We have a short channel solution product. So if you are interested in seeing, just let us know. I can show you. You can also send us an email at consulting@nirx.net, and I'll be happy to help you. Okay, without further ado, so Dr. Hendrik Santosa, please welcome. He's a research instructor at the Department of Radiology at the University of Pittsburgh. His research interests include statistical method, brain-computer interface, hyperscanning, advanced brain signal processing, and multimodal neuroimaging techniques. Dr. Hendrik, very welcome. We are very, very happy to have you here with us today. Thank you very much.
Okay, thank you.
Okay, we can see your screen.
Okay, thank you, Maria and NIRx for inviting me to give a talk in this webinar. So today I'm going to present the recent publications using short channels, using NIRx system also for the multiple comparisons. So my name is Hendrik. So I am part of Hubert Lab. That Hubert is my mentor. And this is the outline. So before going to the methods, I will explain a little bit about the typical fNIRS data, and then do some kind of like literature review, what people done in this algorithm, and then explain the experimental paradigm using the NIRx system, and then also explain the analyzer toolbox. In the end, I will show the demo. So in the result, mostly using ROC analysis, the first result will be show the benefit of using short separations as prefilter versus regression. And also I will show the comparison with the performance comparison between autoregressive and OLS. I will also explain the performance per quality of data for the short separations data. And then in the end, I will explain what is the maximum or the optimum number of the short channels. And I will conclude this talk. And in the end, like 20 minutes, I will show using the toolbox how to use the short separations in GLM. And this paper has been published last year in the neurophotonics. You can go to the website to see in the detail. Oops, sorry. Yeah, for detail equations and all detail informations. And this is the typical NIRS data. You can see a little bit motion artifact, the outlier, little bit spikes, and then you can see, if you zoom, you can see the cardiac around 1 Hertz. You can see also respirations about 0.25 or 0.3 hertz, and also you can see blood pressure, or this is low, like very low, 0.1 hertz frequency. So if you see this one, the physiological noise can be misinterpreted as brain activity and can drive both false positive and false negative estimates. So this error particularly troublesome when there is some possibility task-induced systemic change in the epoch signal. And then this is the previous publications. There are many actually in the publications. So there are two main driven part for this processing. The first one, like if you see, you can do prefiltering approach or you can do statistical approach. So in this study, mostly we use all this algorithm using ROC analysis to see the different performance or compare the sensitivity and specificity of these various publication. This is also include the sort separations using, I will explain after this one. And then the first one, this is prefiltering method. We already know this one. So people use PCA. You can use baseline derived PCA. So it means like you need baseline information. Like for example, you can ask subject 5 minutes to do resting state, and then use that information to get the first component, components. And then because that component is like derived by motion artifact, you can remove that one. Or if you don't have that baseline, you can use also PCA, and also you can use sort separations prefiltering. The first one, like this is the most common prefiltering in the literature. So they just check residual between long distance and short distance. And then in the end, you can do short separation filtering via image reconstruction. So we compare also this one. This is using optical forward model. And then in the second part, there are two options. You can do pre-filtering, or you can do statistical method, or you can combine them. So in the statistical methods, the first one, like you can use sort separation as regression. You can do a GLM, and also the mixed effect. So this mixed effect similar with the sort separations, but the sort separation coefficient will be more statistically informed by other channels. So we put another term in here for the mixed effect models. So this is the experimental paradigm for this study. We have this NIRx system. We got this one as prototype last time. So we have about 20 channel. So 22 channel for long distance and 8 channel for short distance. So you can see the green one. This is the short distance, and this is covered in the motor area. So the center to center between long distance and short distance is about 13 to 15 millimeters. So in the previous publication, people already done the optimum number for the distance between center to center between long distance and short distance should be like 15. So this NIRx system also in that range. So there are 3 different tasks. The first one, resting state, ask subject to do nothing for 5 minutes, and then breath holding. So ask the subject to hold their breath for 25 seconds, and then after that, normal breath for 30 seconds. So this is 2 runs. And then the third one, ask the subject to walking in the treadmill. So we have treadmill in other lab. So we set the treadmill for 5 kilometers per hour. And then the last one, ask subject to imagine walking. So also 5 runs. So the combination of the processing, we have two data sets, breath holding and then resting. So breath holding also, we split also two different parts. This is lock breath holding, and this is random breath holding. I will explain after this one. And then the pre-processing, we just do nothing, the first one, and we use PCA. We use PCA, oxy, deoxy, and oxy and deoxy together, and either oxy and deoxy, and also short separation as filter. And the GLM, mostly we compare two different model, ordinary least square and autoregressive iterative least squares. And also we put also short separation as regressions, and also in the end we also compare with the mixed effects. So this is the data the breath holding looks like. So you can see the blue one. This is the blue. So when a subject hold their breath, you can see this response. This is raw data, and then this is the second one and the third trial. So we have three trials every sessions. And then for random, we jitter this task, not matching with this one. And the last one, panel C, this is the resting state data. Okay, so all the processing using ROC analysis. So in general, this is the ROC looks like. In general, the first figure, this one, this is compare the true positive rate and false positive rate in the ROC curve. The ideal one should be like go to is almost to 1. So the area under the curve should be 1 for the ideal one. And then for the control type 1 error, this is showing the true positive rate against false positive rate. Sorry, this is wrong. So this is should be like p hat compared with the FPR. So the ideal one should be like diagonal slope equal to 1. So that's the ideal one. So if the signals like more than above the diagonal is overestimate, under the diagonal is underestimates. So in the ROC analysis, we get oxy and the oxyhemoglobin after modified with Lambert law, and then put the event. If we have that event, just use that task event, the log event, or if you want to make it random, we can make it random. And then we stimulate using that evoke response in the half of the channels, and then compute the FPR and TPR, and then we repeat a couple of times. So usually we repeat like 10 times for every pipelines. So this is the result looks like. So out of like about 60 pipelines, actually we have more because the pre-processing many for the pre-processing. So let's say we have about 60 pipelines, 3, 4, and 5. So we just only select 7 right now. This is the left figure. This is the area under the curve from the ROC from this one. So against the ideal one should be AUC equal to 1. So bigger AUC is better performance. So if you look to this one, this is short separation as prefilter, or you can use as short separation as regression in the autoregressive. All the figure in here using autoregressive. So the conclusion is autoregressive with short separation is the good one in our study. Actually, for the mixed effect is the best one, but because the processing is 10-fold longer to solve, so we end up, okay, it's not worth it to use that mixed effects. So we just conclude that autoregressive with short separation is enough. And then before, this is the resting data, and this is the breath holding random data, and then this is breath holding log. So as you know that this is breath holding log, it means like this is the worst case scenario in the data. So it means like the systemic noise is co-occurring with the task induced. So this is the breath holding. You can see the performance is lower, but still more than 50%, more than random chance. So the conclusion is short separations improve the performance. As we see this one, without short separation is lower than with short separation. And then the second one, it is better to use short separation channel as regression than prefilter. You can see this is prefilter 3 and 4. Sorry, this is too small maybe. This is, but if you look to the last two one, this is higher than as short separation as prefilter. And then also we look using the table, the specific area under the curve with different pre-processing. Without pre-processing, PCA and baseline PCA. So if you see, if you do nothing without any pre-processing, with short separations, S regression, and autoregressive is the best one. Actually, if you use PCA, increase a little bit more, but it's okay if you have this one. And then if you see this one, if you don't have the short separation channels, you can use baseline PCA as pre-processing for your analysis. So this the first one, resting data, and this is breath holding data. So the conclusion for this one is it is better to use oxyhemoglobin and deoxyhemoglobin together. You can see A and B, for example, this one, A and B. So A is better than B. So it is better to use HbO and oxy and deoxyhemoglobin together rather than or either oxy or deoxyhemoglobin. And then short separation data should be recorded when possible. So if you have NIRx system with the short channel bundles, it's better to use that one to get short separations data. If not, you can use baseline PCA. If you don't have, if your population is kind of like maybe kids populations, you cannot ask subject to do nothing for 5 minutes. You still can use PCA if you want to remove the systemic noise. And then the next question is, it is better to use autoregressive or OLS? So we also compare these two algorithms, autoregressive and OLS, with short separation as regression and mixed effect also. So if you can see from here the type 1 error, the ideal one should be like diagonal slope equal to 1. So OLS, you can see this one has high positive, right? It means like when you have p-value 0.05, it's your OLS report about 0.7 on 0.64. So it's high false positive, right? But when you use autoregressive, you can see this is it's kind of like about 5% is the same one. This is for breath holding data, for random, same one, and also for the breath holding logs. You can see the OLS has like high false positive, right? So again, in here, autoregressive improve control type 1 error. And then what happened when we plot the activation map or the T maps for the breath holding data? So this is using OLS, the upper figure without any pre-process, without short separations, and this is using short separations. So you can see this is kind of like global response. Black it means like not significant. For the color, this is like significant. P less than 0.5 less than 5%. You can see the global response everywhere. So what happened when we use the autoregressive? So you can see the global response reduced using autoregressive even without short separations. But in some channels, in some files, in one subject, this like outlier. So I don't know what happened with the subject. So this is the channel, 22 channel. This is only long distance channel, and this is a NIRS file. Like we have 12 subject and repeat 2 times 2 sessions, so about 24. So in here, we can conclude that autoregressive is performed better to reduce the global response. So what happened when we, because in the ROC we can set the contrast to noise ratio. So contrast to noise ratio for the previous slide, every slide we use 0.7, because in our experience in other data sets, 0.7 is the contrast to noise ratio in our typical study. So the contrast to noise ratio defined as the peak magnitude of evoked response to the standard deviation of the oxy and deoxyhemoglobin. So you can see this is for the 0.7. Autoregressive with short separation regression is still the good one, but it seems like more than 2, because okay. So this is the AUC, because more than 2, it seems like will be like saturated, it will be like IOC equal to 1. So the conclusion for this one is offer the physiological range of between 0.5 and 2. Autoregressive with the short separation channel was the best approach. And then by looking to the quality of the short separation data, we use scalp coupling index. This is from Luca lab, from Polonini labs. So you can look to the paper of Phoebe papers. So the idea is they introduce the idea you can use two different wavelengths data. If the correlation between the two wavelengths is more than 0.8, it means the data is good. So 0.8 is the threshold from them. If less than 0.8, maybe you can remove that one, or this is just only to quantify or to judge whether the data is good or not. So by looking to this SCI, in our data, about more than half of our data, the SCI is more than 0.8. So it's good. So this is the performance when you have this low scalp coupling index, and then this is the performance when you have really high good quality data. You can see this is about almost 0.7. So the next question also, okay, we know from the previous slide, the short separation channel will improve the performance. And then the question is whether we need to use only one short separation, or we need to put short channels in every source positions, because that's the some group also proposed that idea. We need short separation in every source. So we try to answer that questions. If the systemic noise is global, maybe one channel is enough. But not that the case, because the blood vessel and geometry of the head. So this is kind of like not global anymore. So the short channels will be more. Short channel will be benefit for this one. So you can see for resting state data, when you add more short separation channel, this is the nearest short separation channel 1 to 8. When you add more, when the performance or the IOC also increase. Again, the autoregressive is the best one compared with the OLS. For the IOC, of course, for the type 1 error also the best one. We already showed that one before. And then this is for breath holding random data, and then this is breath holding log. So yeah, this is the conclusion. So increasing the number of channels, the performance has slightly increased in the IOC. And again, it is better to use HB oxy and deoxyhemoglobin together, if you see this one, compared with either oxy and deoxy only. And then by looking to the, okay, this is like similar. So we try to compare. This is similar. We try to compare between the performance between pre-filter or regression for the short separation channels, and this is the left for as pre-filter, and this is as we put short separation in the regression, and then this is the nearest from 1 short separation channel to 8. So you can see, but by putting the oxy and deoxyhemoglobin together, the dot one, the performance is better than the solid one. Solid is only to put oxyhemoglobin. So again, it is better to use short separations in GLM compare short separation as pre-filter, and then combining oxy and deoxyhemoglobin for both short separation filter and GLM, slightly better performance than oxy only. And also again, this is similar. By increasing the number of short separation channel, the performance also increase in the AUC. And also we try to process. We try to process. This is all the line. This is from the subject. We have 12 subject, and then 7 data set: resting, breath holding to breath holding, imagine walking to, so 7. So 12 by 7 this is only for short separation channel. So we use SVD from all channels. So it looks like using 5 channels. It looks like using 5 channels is enough in this data set. So the 5 channels explain about this line is about 90% of the variance. So you can say that around 5 channel it can explain 90% of the variance of the data. So it is better to have short separation channel in every source position, but because the system maybe we have limited number of channel, so we still can use the limited short separation channel. Even like in this study, actually 5 is enough. If you compare with the previous figure, also sorry, set back again. So you can see if this is 5 increase, and after 5, okay, slightly increase, but 5 is enough for this study. And then in the end, we try to use bandpass filter in the before the rock analysis. Why we did this one? Because in the literature, mostly people use bandpass filter. They think that because the respiration is about 0.3 hertz, they use low pass filter for 0.5 hertz, and then high pass filter to remove the slow drift. So as you can see, this is oxyhemoglobin data. The dot line, this is the reference without high pass filter. So and this is without low pass filter also for this one. So you can see if it's okay to use high pass filter for this low cutoff frequency, because this cutoff frequency using 0, and this is 0.005, and this is 0.008, and this is 0.016, and so on until 0.064. So it is okay to use high pass filter, but when you use more cutoff frequency, the performance will be decreased. And then there is no benefit using high pass filter in this data. The same thing with the this is low pass filter without low pass filter, and this is with low pass filter 0.5 hertz. So using low pass filter, the performance kind of like increase. You can see in here, this is 0.7 and this is 0.7 more, and there is some benefit using low pass filter, but not using high pass filter. The same thing with the oxyhemoglobin. This is oxy and the oxyhemoglobin. So the conclusion is high pass filter, either with or without short separation regression in the model, has lower performance compared to the GLM alone. So conclusion is the short measurement improve the performance. We can see. So it is better to use short separation as regression than a filter. Mostly in the literature, people in the first like when they know about the short separation, mostly they use short separation as pre-filter, but in this study we saw that it is better to use short separation as regression. And then autoregressive is better than OLS. First one, higher AUC, it means like better performance, and also better control type 1 error. It means like what the MATLAB report and the FPR is good, not overestimates. And then the last one, if you don't have short separations measurements, you can contact NIRx if you have NIRx system. But if you don't have that system, you can use PCA. So it is better to use baseline PCA because you it is better to use baseline PCA. You can ask subject to do nothing for 5 minutes and then use that information to get the first couple components, and then use that one for your task or for the FOX signal. But if you don't have the baseline the resting state data, for example, for kids population, you still can use PCA if you want. And then what we done in this paper, we use FOX signal, and then this is on progress. Praduma working on the comparison with pipelines, the same one using ROC for resting state connectivity. So I think he already finished with the data processing. He's starting to write the manuscript for this study. So hopefully we can see in the next couple of months. And this is just for your informations. This is the same data, not same data. This is the same system using NIRx from Felix lab. So they published this paper in Neurophotonics like 1 week after we published this one. This is kind of like similar. The data set same, the 8 channels, short separations, but the difference is using hand grasping. So the same one, if you see this one using long distance, subject 1, 2, 3 until 5, and this is short separation channel, and this is the regress signal. So it is better to use the short separation as regression. So this is the toolbox. So right now I'm going to show how to use the NIRS toolbox for the short separation channels. You can read this paper if you want to know about the detail for this toolbox, and you can also download from here for the scripts. And this is open source. You can use that one. You can edit in your own local computer, or if you want, you can contribute. You can ask Ted Hartford if you want to contribute. And mostly this is using for fNIRS data, but we can also process a little bit more for EEG, MEG, and fMRI. Okay, so I'm going to show. So Maria, can I show now?
Sorry, Hendrik. Yes,
Yeah.
I can see your I can see your MATLAB screen.
Okay.
Please go on.
Okay, so also this script, you can, if you download the toolbox, you can get this script. You can run this one in your computer. So you just only call fNIRS thing. Yeah, you can call this M file, and it will open this one. So for the like until line 129, this is ROC test. This is ROC test. You can run this. We explain in detail in here, like multiple comparison. Like this is pipelines. You can add more if you want. But for the demo, I will show using the SIM data. So we have SIM data for the short separations. This is the SIMS data. Okay. So you can call this is NIRS testing SIM data for short separations. So you can see if you type probe link. So you can see this is source and detector pair with the type, and then this is the label, short separation label. This is false, true, and this is, so it means this is for the short separation. So the good thing is if you use NIRx system, it will automatically label with this one if you have the short separations. But what happens if you don't have the NIRx system? For example, you use Tekken System or other system. We have these modules to do to label short separation. For example, you can type job. So you can see, you can type this one, and you can see the maximum distance is 15 millimeter. So it's mean if less than 50 millimeter, it will label true for the short separations. So this is, for example, you can load data, for example, other data, not NIRx data, and then you can give the job for the label short separations, and then you can run this one. So in the end, you will get the same things with this one. So you will have the short separations label in the last column, and then after that, we can run this one, the basic pipeline. So the first you need to resample. It's up to you. So we usually resample this one. The default is I think 4 Hertz. Yeah, the default. And then get the optical density, and then using Beer-Lambert law, and then get the hemoglobin data. So you can see this is the hemoglobin data for with short separations. So you can see this still have this one, and you can see also the probe configuration. Oops, another screen. Yeah, you can see this one. This is the short distance for the SIM data. And then, so this is how to use the short separation in GLM. It's very simple. You can run this one, NIRs.modules.glm. You can change this one to your methods if you want. They are autoregressive, OLS, or you can type, I think you can type like this. Yeah, so there are a couple options. You can use OLS, NIRS SPM, autoregressive, this also. So you can see for default, we use autoregressive. And then if you want to use the short separation as regressor, you can change this one to true, like this. So it's mean right now we use short separation as regression of no interest. It means we use that one, put short separation in the regression, and then after that we throw that one. So when we run this one, okay, so we run that one. We have the statistics for the short separations as regressor. So when you compare this one, you have this one, but for the after regressor, you don't have the short separation anymore. This is only long distance, because after we use short separations as regressor, we throw that one. So this is short separation as regressor of no interest. Okay, so it's very simple. Using this function, calling the just give this one to true, and then you will get the same result with the paper. And also what happened, how to use the short separation as pre-filter if you want to do resting state connectivity, for example. This is the function. So this is NIRS module short distance filter. So default the maximum number component is 6. So there are option also split types. So you can select to split type is false. It means no split. We use oxy and the oxyhemoglobin. That is the best one from the paper. But if you want to use only oxy and also only the oxy, you can change this one to true. And then after that, you can run in the oxyhemoglobin. Sorry, for the hemoglobin data, and then this is hemoglobin for the short separations channels using pre-filter. Yeah, this one. So if you run this one from the beginning also, we put one data set in the website, in the GitHub and Bitbucket, so you can look to that information. Sorry, I didn't run this one. Okay, so you can see this is raw. Okay, and then this is the geometry looks like. Oops. Yeah, you can see this is like very close each other. But yeah, this is the short separation. This one you can see in here, and this is the data looks like. Let's see. Okay, so this is breath holding data. This is breath holding data from subject. This is stats data. So you can see this is when I ask that to hold their breath. This is start and this is end. This is start and end. The data looks good. So the limitation of this study is the data is too good because there is no outlier even for the walking task, because the NIRS X cap is good. It's kind of like very tight. So we don't know if we put like crazy motion artifacts in here, but for the previous study also the autoregressive is enough also for removing the motion artifacts. Okay, so this is the data. You can do the same thing, pre-processing, and you can do also the pipelines, GLM with OLS, GLM with autoregressive, and so on. This is pipelines, and you can also plot the HRF response. And the part 2, this is the ROC test. You can look to this one. So I put like one iteration. You can set up to 10 or 100 if you want in your local computer. So in the end, when you draw this one, you will get the same result with the publications. Two different figures when you draw this one. The first one, sensitivity and specificity, and the second one, p hat versus the FPR. Yeah, I think Maria, I think I'm finished with my talk. If you want, go to questions and answer.
Thank you very much, Hendrik. It was really nice talk. We definitely learned a lot. We have some questions. One question, so I think you mentioned just at the end right now, so would be how to use the short separation without in the resting state data, for example, without the GLM. I guess this you said that you should use the filter, right? The filter with
the
short channel.
Yes, if you want, yes, you can I put this one in the toolbox in the demo also. Yeah.
Okay.
In the end, you can use for the because for that resting stats data you cannot do that GLM things. So you can put this one as pre-filter. Exactly. So Ted put that one in advanced.NIRS. Usually we use NIRS.modules, but Ted put that one in advanced folder.
And this is also the one you use it for the paper, right? To do the filtering. Okay.
Yes.
Very interesting. One other question that popped up here is you're talking about combining the HBO and HBR to use in the GLM, but how exactly do you combine this information? How do you do this in the toolbox?
The GLM one. Okay. Oh, okay. Sorry, forget where's that part.
No problem.
Where's that one? Sorry. Okay, so if you put that one as true,
true means that it will use this combination of HBO, HBR. Is that right?
Okay. For default, yes, but we can set that one.
Okay.
Okay, so Okay, so this is add such a percentage regression. Yeah, I think we can set in here. Yeah, I think we can. I forget that one. Yeah, by default is true. Oxy and the oxyhemoglobin. I think last time I just do manually. I think.
Okay.
If
Okay.
want to put like oxy and the oxy only.
Okay.
Yeah, there's no option in here.
Apart from having the option also in the toolbox, we have also Ted in the audience there, and he also add on top of your answer that we can also this combine section that use both time courses from Oxy and the deoxy as regressors in the model, right? So you're not just using Oxy or deoxy, you're actually combining both and using in the GLM as regressors. Okay. Okay.
Yeah, I mean like both oxy and deoxy, or either oxy and deoxy. Yes.
Okay, so you can choose, but what you show in the paper is that when you use the combination both as a regressors, it's actually better.
Better, yes.
Okay, it goes. Okay, thank you very much. Thank you, Ted, as well. Okay, so you also talked about one interesting topic that was the coupling, so the scalp coupling index that you use with the Phoebe. And we had also a question. I guess I'm more answering this one. So how can we ensure good data in the short separation channel? You can also talk about your experience, Hendrik, but from our experience, to ensure a good signal and less noisy short separation channel. So, for example, here I have like a cap with our short channels. So you see the cap. The bundle is here. Short channels are here below the cap, as you can see. And usually, one good idea is to get the cable management inside of the cap, so you don't have the cable of the short channel going into a source, for example, or blocking a detector. So having a good cable management in the inside part of the cap, it's a good idea. And of course, managing the hair out of the short channel, it's another one. So both of these measurements, combining with very good coupling, very good just adjusting the cap really well with the size of the head and the cap, it's also a good solution. But I don't know if you have any other tips, anything that you did that helped.
Usually we also put the shower cap after we finish put the NIRs cap. We put the shower cap. Looks funny when the subject walking in the treadmill using shower cap, but it helps a lot. So it can reduce the room light.
Exactly. So basically just like an over cap, a black shower cap over the montage to help with the system. Yeah, that's a very good tip as well. Thank you.
And also for the short separation, we cannot adjust again, right? Because it's already there. So after put the cap, we cannot do anything.
Yeah.
But from our experience, more than 50% of the data looks more than 0.8 for the SCI. It's okay. Yeah.
Very good. One other question we have here, it's when using the baseline PCA, which component do you use into the GLM as a regressor? It's the first component only for the PCA?
It depends. Actually, in my experience, using 4 to 5 components, but in the paper you can read, we set the threshold 0.8. It means like when the variant explain about 80% 80% variance of the data, we use that component. So we use first couple components, 1, 2, and after that, after 4, 80% of the variance, we stop in that one. Because when we put more, it will reduce the sensitivity will be reduced.
Gotcha. So you don't really set up the number of the regress the components, but how the percentage they can explain from the data. Okay, that's very interesting. Um,
So you can put in here, if you use baseline PCA using toolbox, you can put one component, 5 components, or you can put like 0.8.
okay, okay.
Yeah, you can put 0.8. So you just only use, because 0.8 is from the Maria paper, Maria Francescini paper. It's kind of like long time ago, that like 10 years after, 10 years ago.
Okay.
She explained 80% of the variance for the threshold.
Perfect. Thank you. We have a couple more questions if you're still up to it. So at the beginning of the demo, you used simulated data for short channels. Do you have anything like this in the toolbox version? Or that's a new feature recently added? So the simulated, this short separation example that you put there, this is new in the toolbox, I guess, right? This short separation.
Not too new. I think Ted already uploaded this one a couple of months ago. Yeah.
Okay, so just some months ago. And if you so if you look right now, so if you download now the toolbox, you can have this testing.
This demo also, we published this demo in the toolbox because we want to show to the reviewer last time. So this is already published a couple of months ago.
Perfect, thank you. Oh, that's also another interesting question here. So what happens if you lose the data in one of the short channels? Do you see this as a big issue?
Do you mean the lose, like not a number?
Yeah, for example, I can try to answer that. For example, in our case, if you use our bundles, you have 8 short separations, right? So if you lose the data in one of them, and since you show that 5 short separation would be already good enough to get a nice nice prediction. So if you lose maybe one channel or the other, wouldn't be a big issue, right?
Yes. And also, I think in the NIRx, I never see the experience showing not a number of data for the NIRx. So I think it should be okay. If loose, it means like just noise for the channels, but you still can use that data.
Okay, and the toolbox would be able to deal with this, right?
Yes.
Okay, good. Okay, I think I will just do one more question, and then we can get, if the other questions that were not answered, we can of course send you later, and our support team will be happy to follow up with everyone. Just for your one last question, so you already shared the online data set for this paper, I guess, but could you is there a difference between the short distance at 10 Hertz compared to 1 Hertz, for example? Do you see any difference that could be in the sampling rate would make any difference from this short channel perspective?
I think yes, but I don't know. I don't know how to compare that one, because from the NIRx we have 10 Hertz. It's like default. And then after that, we always resample that one to 4.
To 4 Hertz.
Yeah, I don't know if there's someone asked 1 Hertz. This is from other data sets or after resample.
And in the data set, it's available, right? If someone would like to do this kind of. Okay.
In the beginning of this demo, it will download that one data set.
Okay. Great. I think that's it for today, Hendrik. Thank you very much for the full presentation and for showing us the toolbox. Again, this will be available on our website in maybe by the end of the week. And thank you very much for everyone that's watching us. We'll see you on the next webinar. Thank you. Thank you again, Hendrik.
Thank you.